Welcome all participants and speakers of our virtual symposium, Functional and Clinical Relevance of Microbiome Signatures. We're just gathering after this, I think, fantastic meeting, 3 days in a row, different subjects, and we wanted to take the chance now to discuss here in this little round of people what the hallmarks are, where the challenges are, what the solutions might be. I would briefly introduce myself. I'm Dirk Haller. I'm a professor of nutrition immunology here at the Technical University of Munich, and I'm the spokesperson of this collaborative research center, CRC 1371. And I would hand over to all the others to make a short introduction. I start with Melanie to my right.
So my name is Melanie Stürmer, and I'm an Immunuta group leader here at the Technical University of Munich.
My name is Markus Turtstadler, and I'm also working at the Technical University of Munich at the Translational and Cancer Research Center.
So my name is Bärbel Stecher, and I'm a microbiologist, and I'm leading a lab focusing on microbiota pathogen interactions at the LMU Munich.
Super. I mean, we're just coming out of this virtual meeting. Yeah, we had 3 days of exciting talks, a lineup of exceptional international experts. And I just see that also an advisor, colleague, and a real expert on microbe-host interactions, Thad Starpenbeck, is trying to kick in from Cleveland. We're waiting a little bit until he is ready to go. Hi, Thad.
Hi.
Fantastic to see you. You're just in time. We're having the warm-up discussion, and then we try to get different points and discussion points through the round and also to you. I'm just saying we had these 3 days of meeting, 3 sections: infections, immune-mediated chronic inflammation, and cancer. And in the inflammation was in the gut, but also in systemic with autoimmunity and neurodegenerative diseases. So if I would ask you guys, when we look at the 3 categories of diseases, and if we ask ourselves, where are we at the cutting edge level in microbiome research? Are there commonalities between the different diseases, or do we have to make really distinct alterations between them? What do you think? Go ahead.
I think it's very much connected. We had infections, inflammation, and cancer. And also the disease entities are very much connected. So, and also, of course, the microbiota profiles and also this distinct species are connected with each other.
Yeah, I think that, so for example, in infection, we study really the interaction between pathogens and the microbiota for a long time. So this is probably one of the disease conditions which has been recognized for a very long time to be connected to the microbiome. And because also pathogens really directly interact with the microbiota. So I think because this was also the opening session, and it already showed that many principles that we observe in infection, they might also apply to other diseases.
What was the hallmark for you of the whole day of infectious and microbiome research?
Well, there were several hallmarks, I think. So one thing that we observed was, or that I noted, is actually that clinical studies, prospective clinical studies, are really important to link specific microbiome signatures also to certain diseases. And in this case, there was a brilliant talk by Debbie Bogart, and she really introduced a prospective clinical cohort of children where she could actually look at viral signatures later on and connect really the infection risk to lower respiratory tract infection, also to changes in the microbiome very early on. I think this was also seen or also introduced in the talk from John Penders, who had a cohort of travelers where he actually looked at the microbiome before travel and then after travel, and then he could really dissect different or identify specific protective structures, for example, in the microbiome that are associated with protection from acquiring antibiotic-resistant bacteria, for example.
If I can ask you this one question. I mean, we heard Andreas Peschel talking about nose microbiome, and I think, at least for me, fantastic was that the chemical diversity produced from the commensal environment in the nose acting against pathogens is enormous. And he worked 6 years on identifying antimicrobial structures. So how far away are we? Let's say in the field of infection, we come to the other diseases a little later on, from designing specific drug applications related to the microbiome. Any idea?
So I think that the work that Andreas really introduced that is really outstanding, where he identified a protective bacterium, Staphylococcus lugdunensis, that is associated with those individuals that do not carry Staphylococcus aureus in the nose. And from there, he could identify this lugdunin component and show that this acts really as an antimicrobial and leads to killing of Staph aureus in the nose. And actually, he is also developing really a microbiota target or a pathogen targeted therapy from this. And I think this is a great example how these identifying really the molecular mechanisms behind these interactions can then also lead to developing specific therapies that would allow us to go away or to go to more specific treatments than with these broad spectrum antibiotics.
I mean, what struck me is the question colonization resistance. Yeah, and you argue on one component acting on one infectious organism. You know, if I would take Melanie into the equation, who tries to identify complexity based on the sequence data available at the moment. So colonization resistance, yeah, what does that mean? Is this at the end important, or do we only have to shoot away certain bugs? If it's colonization resistance in complexity, networks of bacteria, metabolites produced. So what to do?
I think it's still really important which microbiomes are there to start with. And we saw that in Eric Ahm's talk, right? If you give FMT, donor and recipient kind of change the whole setting. So it depends on which microbes are there in terms of which ones engrafts, but it also depends on which ones you put into the system. And I think his kind of end message there was also there's a lot more that we have to learn in terms of like choosing also specific donors for specific recipient to make that engraftment happen as well. So kind of going towards that colonization resistance, but like who is there kind of depends on who can come in. So, and I think that kind of plays into like some of those medications that we give as well that kind of wipe out those microbes. We don't realize yet to which extent we open up the doors for then pathogens, going back to infections, to kind of change the whole colonization patterns that we see in these patients.
I would like to use the chance now to connect that into this discussion. I mean, that maybe you can introduce yourself briefly. You have been here for a week with us. You're a great advisor. You have plenty of ideas. You gave a fantastic talk on fungal interactions on the barrier. So all of that happens somewhere at the barrier. And the barrier, I think, is the connecting glue between infections, which escalate at some point and progress, and then inflammation and also cancer. So barrier epithelial environment is very important. So what's your take? Maybe you introduce yourself first.
Sure. Yeah, so I'm Thad Stappenbeck. I'm the chair of inflammation immunity at Cleveland Clinic. And yeah, I've been, as Dirk mentioned, I've been very interested in host microbial interactions at barrier surfaces for a long time. So there are a lot of great themes that came out of these talks. The talks, by the way, the talks were amazing in this symposium. It was really the breadth and depth of the work that's going on here to me is quite impressive. And to see this kind of all under one roof is really a testament to everyone that's part of this. And then, you know, with a few additional outside speakers, I think it was really great session. But I think the themes here I think the thing that I'm struck by is so there's a lot of things that I agree with that have been said, that really clinical cohorts I think are really critical. I think starting with human studies I think will be incredibly incredibly valuable. And then getting down to what I was impressed with was it wasn't just a lot of sequencing studies, that there was a lot of functional work that was done here. So I think moving into functional studies where you're getting down into specific details, to me, is quite impressive. And to see this happening in many areas kind of across the board, really bodes well for our field, because I think this has been the wrap on the microbiome field for a while. It's been too descriptive. The question is, as we begin to uncover some of these really specific interactions, what the challenge is going to be to convince pharma that this is actually something that's worth their time. They've been very, very resistant to engage in these types of developing these types of drugs. And I think this is something that's maybe going to require someone to do it in a small company or something like that to get this bootstrapped, because this really is not the pharma playbook. And these we're really discovering, I think, really new kind of concepts here and new interactions that they've never thought of targeting.
Well, if I can briefly interrupt you and ask the question, I mean, when we talk about complexity, colonization resistance, which is obviously a complex network of microbial communities in a niche, if we think about therapy, I mean, the one bug therapy is probably easy, but the expectation, I don't know. What can we expect from that? There were probiotics in the first line 2 decades ago, and I think they have just in part delivered what the hope was for. So now complex communities, we had a talk on fecal microbiota transplantation. Where are we there? What's your take?
Yeah, so my take is I think we're getting down to really nice stories of function, and that what has to come out of all of this is we need to understand the rules. So there are overlying principles here that govern all of these interactions that we don't know what they are yet. And I think this is what we're struggling with. So we use concepts that we have a vague understanding of, like colonization resistance, to try and framework this stuff. But I think we're going to have to. I think there's a lot of big discoveries that are yet to be made in the field to synthesize a lot of this work and to compartmentalize it and to crystallize it into real targets. And for me, I think this is where the real excitement is in this field over the next decade, is being able to, as a community, being able to crystallize these features. And we have such great tools to do it. Infection is great. Inflammation is great. Tumors are great. So these are 3 really powerful tools we have to understand the rules. But to me, this is where we have to be headed. Is this?
Well, this was a nice introduction, actually, to the vision of our collaborative research center, because we picked infection, we picked graft versus host disease, we picked inflammatory bowel disease, and we picked cancer. All of them are continuations of diseases in the intestine. And the question is, common rules or completely separate? And I would like to pass over to Markus, this great talk on one of our PhD students, H. pylori infection in the stomach and consequences in the large intestine. So we were talking about immune activations, Th17, but it's a far distance. And so what can we learn from these conditions?
So it's a very good question. So I agree. So with this complexity, we just come now actually into function. There are daily more mechanisms out there. So I think it's not just restricted to one side of the organ where we can just ignore all the rest. So everything is kind of connected. So this oral to stomach, stomach to intestine, and also periphery. So we have seen that the microbiota influences. So if it's coming from top to down or down to top, so that is influencing multiple disease entities and not just in these few organs.
So what's your take? One common rule or different rules depending on context? I mean, this is obviously the debate, yeah? I mean,
I think, as always in biology, there's not just one rule. There are always exceptions, and I think it's not easy to say that there's one common mechanism that leads to all the diseases. There are multiple factors, multiple bacteria, multiple other components of the microbiota that actually convey produce metabolites, other functions of how they penetrate the tissue. So there's tissue-specific functions, but also translocation. So in the end, there are more things to consider. It's not that.
Can I ask one thing? Thad mentioned this compartmentalization. I mean, we heard Cindy Sears talk on biofilms. This is one compartment, which is probably completely different from the luminal environment. Then we talk about a stomach infection. So, complexity, context.
Yeah, when I can jump in here. So I think what we have seen also in several presentations or heard in several presentations is actually that the environment is very important in shaping the microbiome and shaping its composition, but also shaping the structural relationship and the interaction between the microbes. And for example, also regarding this presentation by Anna Ralser, she showed that actually Helicobacter infection in the stomach can create an inflammatory environment in the colon, and this changes the composition of the microbiota in the colon to be more tumorigenic. And she showed that even when transplanting this sort of inflammation-conditioned microbiota into germ-free mice, then it was still kept this more tumorigenic function. And we have the same or a similar, as I think, a similar principle seems to be the case in post-infectious IBS, where travelers returning from other countries that have a higher risk to get common gastrointestinal infections, they also see or get changes in their microbiota, and then they are more prone to develop this IBS. So I think that this conditioning is very important to take into account, and really interpreting also the relevance of the relevance and also the mechanisms behind these signatures. And from the field of infections, we know, of course, that inflammation really changes the environment in the gut. For example, there's more oxygen, there are more antimicrobial factors, and pathogens have sort of learned to thrive in this environment. So they, as we heard from Manuela Raffatello, for example, they produce siderophores, they acquire iron, and these are strategies by the pathogen, and certainly also by certain other symbionts that are enriched in this environment.
Can I ask you one thing? I mean, one reason for me to study commensals was always they're much more interesting because it's not so black and white. You talk about pathogens, they're equipped, they have virulence factors. When you look at the different talks, for example, enterococci, they kind of come along the right in many pathological conditions. So if I would ask you, virulence factors. If I look at enterococci, they have virulence factors which you probably would find in staphylococci and under other pathogenic organisms. They may not necessarily use them. Some may jump over the edge, like the Enterococcus gallinarum, for example, which obviously translocated, goes to the liver, has something to do with autoimmunity. So how do we get a hand on these virulence capacities, and how would we be able to make interpretations of whether or not they are implemented in processes in diseases?
I think there are a few different points that would be important in the context, and I'm totally on the same page with Bärbel there. I think one of the really nice takeaways is how important it is to consider the environment that those microbes live in, and especially if we take them out of their environment and we test them in the lab later. That is a completely different environment. So we kind of need to start either mimicking the conditions or kind of look at different conditions and see how they change their things. And there we saw a really nice talk on the first day as well, where the speaker also mentioned that there is prophages that get induced during inflammatory conditions. And I think that's one of the overarching things with a lot of these diseases as well, that inflammation changes the behavior of those microbes. I think there are a lot of opportunities from the computational side that we can take there as well.
That's what I wanted to hear from you.
I'm getting there.
Expression analysis, no?
Exactly. So we're just there's been so much important work going on in terms of like looking at taxonomic composition and seeing how those microbes change in terms of abundance. But we're starting to realize more and more how important it is to take the environment and their behavior into account, which can change in certain conditions. So if we look at things like metatranscriptomics, gene expression, or the metabolomics, which metabolites they're actually producing, I think that gives a lot more kind of context that is really specific to disease and important to get down to those mechanisms.
Here's the challenge for you, computational biologist. How easy at the moment is it to analyze complex transcriptional, meta-transcriptional data from the sampling to the processing to the bioinformatic analysis? I mean, I haven't seen any of those data, surprisingly, really in a complex environment. So obviously, from this elaborate round of experts, nobody really uses this as a routine. So where are we there?
I mean, one of the drawbacks of metatranscriptomics is it's not the easiest thing to sample. We know that from any type of RNA-seq experiments that we have done already, because RNA is just not as stable as DNA. So if you get those samples from the patient, and a lot of them are home collection methods, you have to be really careful in terms of how they are collected to keep that RNA stable. But once you have a good quality sample and you start sequencing that, you need a little bit more sequencing depth than with the with the metagenomic data, but it's nice to combine these two data types and actually check which genes are expressed and what those microbes are doing in their environment and how this expression changes in disease as well, or for example, in connection with a specific drug.
I mean, one very big focus of our collaborative research center is metabolite analysis, targeted, untargeted. If I look at the presentation from your PhD, Bärbel, I mean, you go in two different directions and pick up some of those thoughts. Number one, that you go into minimal consortia, you combine different strains and then analyze their metabolic capacity in different combinations. So synthetic consortia and understanding their metabolic activity in different contexts, is that the way to go?
Yes, certainly. So what it was a bit surprising to us, actually, the work that Anna Weiss presented on the first day in the first session, where we actually have we got actually, and that's analysis from the wrong side, because we studied this synthetic community in mice for like 10 years, and now we decided that it's time really to also look really at the species and their metabolism and their interactions in vitro. And what we observed is that the whole picture gets much more complicated, and it's very difficult actually to reproduce the interactions that we see in vivo in vitro. And we have to, of course, in the end, we want to avoid really the use of animal models in the end. We want to be able to transfer conditions that are in vivo relevant also to in vitro culture conditions. And I think from really understanding what is the actual in vivo environment in the gut that is relevant, we are really far away.
Yeah.
And that was actually a bit unexpected to me.
So I would like to bring back the host, because, you know, we have different disciplines here. We have microbiologists, we have geneticists, we have computational experts, then we have experts on different diseases, models, and so on. So Thad, to you, what type of model conditions do we need? I mean, we saw. I mean, obviously, the goal is we want to go to the patients. We want to have drugs which work. On the way there, we need to have models. Yeah, we saw pig models, which we developed here in the Collaborative Research Center. There are organoids. The question is the epithelium important? Can we model it? How do we get the immune system there into the equation? How sophisticated are we here?
Yeah, that's a great question. I think as far as in vitro, so in vitro systems looking, say, at epithelial systems, I think the outlook there is fairly bright. There are a lot of opportunities here, I think, to design experimental systems to test your questions. So say you have a specific microbe or specific metabolite or something where you want to look at host interactions. It's very possible to do this now with both mouse systems, human cells, to do this in a way that the differentiation state can be very much controlled. And I think this is going to be very important for the field moving forward. So I think that the key is and there's no one system that's the best system. It's the system, I think, that matches your question, that allows you to test it. But there's a lot of tools that are out there.
Yeah.
There are a number of areas for growth here that I think are going to be quite important. Some are going to be these flow systems, or these what are called gut-on-a-chip systems that are still in their infancy, but show a lot of potential. The ability to adjust concentrations of oxygen or pH, that sort of thing, look at interactions of multiple microbes in a flow system, I think will also be is quite exciting to me.
So would you agree that there are no good or bad models, just good or bad questions or interpretations?
Yeah, I think it's these are all about taking your questions. That's why I love starting with human and starting with really clinical questions. These are always, to me, the strongest projects. And then you drill down and use these in vitro systems to test hypotheses that you can go back to in your cohorts.
I mean, the intriguing thing is that cellular resolution is something the host field is approaching since quite a while. The technologies are there. Now, cellular resolution of complex microbial ecosystems. Are we there? I mean, yes, we can fish out IgA-coated bacteria, but so are we at the single cell level, understanding single cells in a community structure?
I think we're only starting. So I think they're the first studies where they're looking at single cell metagenomic sequencing. I don't think we're at the point where it's widely kind of applicable yet, but it's definitely promising. And I think eventually we do want to go down to that level.
Yeah, I agree. I mean, when I can bridge now to a little other topic, which I think comes up again and again, and I see this on different conferences, that's diet, for example. So metabolism and diet is very close. The rules of diet and the impact of diet on communities, I think, is completely not understood. We saw the talk of Debbie, for example, where you can interfere with exclusive enteral nutrition, which is devoid of fiber, that you can really reduce inflammatory responses and disease activity in patients. Now, the big question is diet acting on the host? Is diet acting via microbial communities? I think she showed some good evidence that it acts directly on microbiota, but how do we bring in this other big environmental component, diet?
Maybe just to add briefly to the EEN therapy, what I always find fascinating is that diet can have as big of an impact as some of those really heavy immune suppressing drugs. But at the same time, it only works in Crohn's. It doesn't quite work in ulcerative colitis. It works in kids, but not quite in adults. And I think it only works the first time around as well, and not quite as well the second time around. So it's this puzzling scenario where it works really well if you apply it early, before kind of any other medication gets in, but only in certain contexts.
Absolutely. So that brings again back the question: context, timing, and the potential of diet. I mean, we heard that the metabolic environment of the host would render infectious conditions in chronic inflammation, for example, or cancer. Do you think metabolism plays a role? I mean, we have obesity as a strong influencer, and
of course,
I mean, comorbidity of cancer.
I mean, diet is a good example how complex it is. In the end, you give what you give as food to the microbiota. Of course, this changes massively everything, metabolism, but also community structures. Maybe also the niches change, and then when the niches change, then also different microbiota to microbe to microbe interaction occur. Then depending on the state or the disease entity, so for example, if there's leaky barrier or leaky gut, as often referred to, then this can also have immense influence. And I think we are only at the very beginning, as many already said, to really understand what is happening there. But I'm now in the field of about 10 years. I started as a PhD student working with microbiota, and what I've seen in the last, even the last few years, what mechanisms we found and how complex this is, I think that has changed dramatically over the past few years. And I think in the next few years we will get a much more better understanding on the types of questions you just answered us. Yeah.
I mean, the people who know me, and I'm in this field now more than 20 years, I'm a big enthusiast. Yeah. When I look at the individual variation, it kind of hammers on me, yeah, because I mean the diversity we see in individuals and how we have to make sense out of the data in a stratified, individual, personalized level. So that could, on the other hand, be a big chance for personalized intervention, because if we can understand rules, then we can apply the rules to individual start points of microbial ecosystems. So.
But I think what you just kind of elaborated on is like just emphasizes how important it is to get to the mechanisms. It doesn't matter necessarily if some microbes go up or down. If we don't understand the mechanisms, we can't interfere. And I don't think, like even with diseases where we know that they're really heterogeneous, such as IBD, I don't think it's going to be a one person, one treatment type of thing, but groups of people where certain mechanisms are failing or in place to kind of cause this inflammation in the disease itself. And you need to figure out what the mechanism is, detect it, and then counteract. So I think that gets us back to that functional side and how important it is to look at that now.
Marcus.
Exactly in alignment with all these mechanisms, and I think there are still thousands, if not even millions of different mechanisms. But in the end, so the topic of this last conference, this was functional and clinical relevance of the microbiome, and we need to understand the mechanisms. Then we also have functional and clinical relevance that we have to understand. Because
I mean, it seems that there are global mechanisms acting on the microbiome. If you listen to this industrialization impact on diversity, for example, is it really so that we lose certain capacities in the microbial environment in the gut, which we potentially never get back if we not transplant it back, for example?
Yeah, I mean, this is really disturbing that we see this trend now that the diversity of the microbiota goes down, and that industrialization plays a big role. And of course, because you mentioned already diet, so processed diet, less fiber-rich diet, for example, is known to play a role there. But besides, there's drug intake. There are also other family size and also other living conditions that are very important. And I think we now also Eric Alm really introduced this microbiome conservatory project where this is really aiming at trying to really conserve the diversity that we still have. Although we have no idea whether this would be useful in 10 or 20 years, because we don't know whether this really plays a role, and we should really have a more diverse microbiome now, at least, and we are able to study this in more detail, and then later on come back to these repositories and develop new drugs and therapeutics.
I mean, the puzzling thing still is cause-consequence. And if you look at this, that a global mechanism runs over generations, and then we all sit of certain host susceptibility genes and develop certain diseases. And now the real question is, still, not everybody who is susceptible from the host side would get the disease, not even in the same environment. Anybody of you suggestions how we could address this, like the early onset where not the disease overrides already many of the effects? So we often get samples during a disease. It's very hard to get prospective samples in a controlled fashion. Large cohorts are necessary. So how do we address this? Thed, any idea?
Wow, this is like the hardest question that you've asked. So I mean, because if you look at you look at what's happened in genetics, like how many, I mean, what are we up to now? Like hundreds of thousands of patients in IBD, just IBD, that have had whole exome sequencing, and how far has that really gotten us with just genetics? Now you want to do the same thing for environment. where we want to do deep dive and collect all this data. If you look at this, kind of the back of the envelope calculations here, again, you're talking about hundreds of thousands of patients. Then we'll have a better idea of environmental effects. Even though we think we already know this, but I'm not sure that we do. Then you want to take, these hundreds of thousands of patients here and these hundreds of thousands of patients here and try and overlay them. I think it's going to be statistically really hard to do this. So I think we're going to have to come up with other ways to try and get to ideas where we can really start testing things and experimental systems where we can get to this faster. This is, so if you look for the number of gene plus environment studies that are in the literature right now, there are very, very few, because this has to be stuff where you're pretty certain of it going in before you can test it. So this is the challenge.
So we have this very cool project in our collaborative research center. Niels Hoffart presented on that, systemic allergic inflammation. And there you have tools to measure actually the immune deviation by measuring actually the seroconversion or antibodies, which or the sensitization in allergy. We have seen this in type 1 diabetes with the seroconversion. So there are the effect on the immune system first, and then either the decision is disease or no disease. That could be those types of diseases and models could be something we could use to kind of get closer to the question of cause and consequences also in a human setting. Treatment naive IBD patients is maybe something else we could address. Is there anything out there as well?
That's hard because about this GEM study, right, in IBD. So this is looking at studying our brothers and sisters of patients that have developed inflammatory bowel disease, because a larger proportion of these kids will develop IBD. So that the key is you can learn something very early on about what are some of the early factors. And they have done really deep dives into serum, into stool, into urine, and it's been a slog. I mean, it's you realize how hard it is to do this. Well,
I guess that means we need to go back to the models. We need to find or use good models, use synthetic consortia, which are hypothesis driven, reflecting certain conditions. We need good tools and methods. I think that's what we need to do. I mean, if I can wrap up this here a little bit.
Can I say one more thing, Dirk?
Sure. Go ahead.
Because I think the last piece, and you and I have talked about this, as I think we have to get back to physiology as well, too.
Yeah.
And I think we have to pay attention to what these perturbations do in just the normal host. And I think this is where the rule finding can happen. You wonder about, and we need some creative solutions to this, like with microbiome. So is what's going on in the microbiome, are these microbe interactions, say in the duodenum, and the metabolism that's affected there, do the resulting metabolites not only affect locally the host, but do they affect things downstream in the gut, since it's a continuous system?
Yeah.
And are we not analyzing our in vivo models properly, and we're not paying attention? We tend to pay attention to what's going on in the poop, but I don't know that we necessarily are thinking upstream and thinking about the physiology, not only of the whole intestine, then, but potentially the whole animal.
I mean, we were talking about the colon, the unknown organ. Yeah, I mean, we know, I think, a good deal about the small intestine, also from the anatomical physiological point of view with motility. If you look into the colon, where bacteria continuously grow, where food remains 40, 50, 60 hours, where slow motion takes place, water is not equally distributed. I think completely different rules apply there for this ecosystem. I fully agree. Physiology comes back. We probably have to educate a whole new generation of scientists, because we lost the physiologists along the way, I guess. I'm not sure.
We competed them out. Exactly.
If I can add one more thing as
Sure,
well.
go ahead.
I think there are also like two separate questions I think that we need to tackle differently. One is disease onset. So what's the cause why the system kind of
Yeah.
tips and causes disease to actually start? And then the other thing are the disease mechanisms. And I think a lot of the cohorts where there's established disease, we can look for mechanisms and actually new treatment options. The disease onset question is a lot harder because you would need large prospective cohort studies actually
Exactly.
figure out when those patients start getting symptoms, ideally get a sample before and after, and that's where it gets really hard with the human cohorts. But I think on the mechanism side, to come up with therapies, or human cohorts can actually be really helpful in terms of giving us those answers.
I think at the end we need to go into interventions, try things. And I mean, we had this one talk on fecal transplantation. Many people say that's very crude. Yeah, it is crude, but I think it's one of the controlled interventions we can do at the moment, where we, at least to some extent, modulate existing communities with something which we try to engraft. So maybe Bärbel, one last statement from your side. I mean, you're a basic microbiologist in infection. Where is the future for therapy? I mean,
Well, as you said, I mean, we're starting usually with patient cohorts, and we go to mouse models or in vitro models, and we try to dissect the mechanisms. And from there, we can then try to really develop specific and sophisticated tools for specific interventions in a certain disease context. So for example, I think one example that I would really like to highlight is this method that was described by Manuela Rafatello that is based really on such an observation. So we observe blooms of Enterobacteriaceae in different disease conditions, ranging from infectious diseases, from antibiotic treatment, to IBD or cancer. And it's really a common underlying mechanism why these bacteria can actually thrive under these conditions. And that's why, because they are very, very good really at producing siderophores and scavenging iron. And then she showed that in the IBD, in the infection condition and also in the IBD context, vaccinating the animals against enterochelin actually led to specific reduction of Enterobacteriaceae blooms. And this, I think, is a very nice story that in the end comes to a very specific therapy.
That would argue for commonalities between diseases, and that would be much easier than to develop kind of therapeutic applications.
I mean, as I said, I think infection, so we started with infections, and now we see that pathogens may even play a role also in the induction of colorectal cancer. So there is Clostridium difficile or Clostridioides difficile that is associated and is really causally involved in promoting tumor growth in mouse models. And there are probably also other pathogens or even bacteria that produce certain virulence factors that are not actually meant to cause harm to the host. They're just actually there because they provide the bacteria with a competitive edge against the other symbionts in the gut. And I think there we can really learn a lot also from infection and from looking at pathogens and how they do it.
I'm fully with you. I mean, this is why we chose here in this consortium to use the 3 different disease conditions to see a little bit beyond the individual disease of whether there is commonalities or whether there is real disease-specific mechanisms, which certainly is true for some. I want to touch on one last thing. Methodology, because we heard this in this one talk from Gabe, that methodology can change the way we look at things. So, Melanie, I mean, you work with a lot of data. Thad mentioned that there are huge data sets out there. We need even more larger cohorts. People do things. Are we there in terms of methodological harmonization? What is really required to get to the next level if we want to compare globally data?
So I think there's still a lot of work left to do, like in terms of coming up with standards that are widely accepted across different countries. there's still a lot we have to do, and also come up with ways to agree on how to collect samples. I think from the analysis side, there's always going to be different ways and kind of disadvantages and advantages to one way or the other. In terms of collecting and producing the data, if we don't agree on kind of a common way to do that, it's going to be really hard to compare them. In terms of the data sets that we have out there, they do have all of these biases in there, which is okay. You have to start somewhere, and you have to also figure out where those biases are. I think where they're still really useful, if you identify a mechanism or a very strong signal in one data set, you can still check if it's present in another data set. If it's not there, it's a little bit harder to tell whether it's then true or not, because it might be due to those biases that are involved. But I think in terms of replication cohort, we can still use different data sets that are out there and produced in different ways.
I mean, one way to learn is sometimes that we see differences which we don't understand. I just remind us on this talk from our group where the XIAP mice were compared in different locations. In one location there is disease, in the other one there is not, and then you work out a certain mechanism. So even that requires some sort of standardization. If people see one effect in a mouse model, I mean, you can comment on this. You just analyzed in genetic variants of cancer the different microbial communities. So
yeah,
I think
it's what Melanie said. It's very important that there needs something be a standard that studies can be compared, and not just data sets, but also different studies with preclinical models. Because if in one animal facility you find this part mechanisms and it doesn't hold true for other animal facilities, so where does this take us, right? So I think there needs also be standardization and to see what can be translated to a very common mechanism, also to human situation, and not that stays in one animal room and is basically.
Well, I think the use of these oligomicrobiota or mouse microbiota you established as one example. Other synthetic consortia could be used.
But on the other hand, I think, I mean, I agree with you that standardization is for some studies is really important. On the other hand, if we had standardized our microbiota 50 years ago in different animal facilities, we probably would not have discovered many disease associations with the microbiome. When people moved their specific mouse model from one facility to the other, it got rederived, got a new microbiota, and suddenly the phenotype was gone, or there was a phenotype coming up. And that's why we started really to look at really the role of the microbiota in cancer, for example. That started all with mouse models.
But I also think, like, in terms of these things, it's important to distinguish between biological kind of variation and technical variation. Sure. And I think getting rid of the technical variation is probably a lot more important than the biological variation, because that can give us additional insight with, for example, mouse models where the microbiome just gave different results all of a sudden. So I think it's important to distinguish between the two.
And also keep in mind, then working with mice, it's not basically a physiological microbiota. When we talk about human microbiota, it's very standardized in mouse models, so it might not be harbor all the relevant pathogens or not. It's definitely not harbor all the pathogens and pathobionts that we are discussing now here, and what may lead to.
I think this is a good closing word. Thad, you want to add some?
Oh boy, we could go on all day about this. We had a lot of conversations in the bar about this, about whether we should standardize this or not. I mean, because we've certainly, me and many other people have discovered things that in the non-standard microbiome that have been very important. But at the same time, it would be nice to conduct some experiments where it is absolutely refined. So again, I think it's your experimental system in the question.
That's exactly what I think. And now, really, as a closing remark, I think we need diversity in science as we need diversity in microbes, but we need to know what we're doing. And for that, we need to know the rules, and for that, we need clearly standardized ways to look at. So I think we're on the right track. I would close this lively discussion. I think we now discussed more than an hour or so, because people should look at it. So we are chronic enthusiasts. I think that's very clear. We love what we do. And I think what we saw the last days is exciting. And I think we, with our questions, if we reflect what we heard, I think we're on the right track. We all go for the best and hunt the microbes we think are important for health and disease. And I think we close this round. We wish everybody a good evening. For us, it's now 8:30. We already have a long day. That is in Cleveland. You start your day. Thanks for joining that. That was wonderful. See you soon.